Hello everyone, and welcome to this Gen webinar titled Specificity Testing of Antibody and CAR T-Cell Therapies for IND using the Membrane Proteome Array. This webinar is made possible through sponsorship by Integral Molecular. Extensive specificity analysis is essential for successful drug development and a safety requirement for antibody-based therapies like CAR T entering IND. Tissue cross-reactivity studies have traditionally been used to screen for off-target binding. However, their predictive value for in vivo safety and toxicity is poor. For this reason, Integral Molecular created the Membrane Proteome Array platform to reduce the risk of antibody-based therapies by testing specificity across the entire human membrane proteome expressed in live cells. This technology has emerged as the leading solution for antibody specificity profiling, utilizing sensitive high-throughput flow cytometry to identify binding interactions on a protein level. In this Gen webinar, our panelists will talk about how they use the Membrane Proteome Array platform to quickly identify potential off-target interactions, resulting in successful IND filings for CAR T-cell therapies where traditional approaches failed. They will also discuss the latest additions to the 6,000 protein cell array, as well as the FDA's consideration of it as a qualified drug development tool. My name is Jonathan Grinstein, Senior Editor at Genetic Engineering and Biotechnology News, a Marianne Liebert magazine that has been avidly covering the biotech industry for more than 40 years. With a focus on specificity screening, this webinar assembles experts in antibody discovery and development and features presentations from Integral Molecular, Boletta Bio, and the University of Pennsylvania. Our speakers today are Rachel Fong, Director of Sales and Alliances at Integral Molecular, Darshil Patel, PhD, Associate Director of Preclinical Research at Boletta Bio, and Donald Siegel, MD, PhD, Professor of Pathology and Laboratory Medicine, also the Director at the Division of Transfusion Medicine and Therapeutic Pathology, and the Director of Center for Advanced Cellular Therapies at Perelman School of Medicine at the University of Pennsylvania. This is a reminder to stay tuned for the Q&A today after the presentation. All right, let's get started.
Hello, everyone. Thank you so much for joining our webinar today. My name is Rachel Fong, and I am the Director of Sales and Alliances at Integral Molecular. Today, I would like to take some time to tell you about specificity testing of antibodies, bispecifics, and CAR T therapeutics for IND using the Membrane Proteome Array. As many of us are aware, therapeutic drug development is an extraordinarily long process requiring a huge amount of resources and has many challenges along the way. One of those challenges is ensuring the specificity of the molecule. Antibodies have been known to exhibit polyspecificity, and we have seen this be the case at Integral Molecular, where about 25% of the antibodies we screen for customers exhibit off-target binding. Off-target interactions are concerning because they can potentially lead to severe adverse events or major side effects once they enter into the clinic. Our goal is to aid drug developers by de-risking their candidates during the drug development process. In order to do so, Integral Molecular developed the Membrane Proteome Array, and it is the leading solution for antibody specificity profiling. There are a number of reasons why this is the case, and I will discuss each of these in more detail today. I think the best validation of the MPA, though, is the fact that we are trusted by over 100 companies who have repeatedly come back to us to test their molecules on the array. Many of these have actually integrated the MPA into their development pipeline to be used as a standard as they make their decisions moving forward. We have a lot of customers who have had successful IND submissions using the MPA data, and a number of them have actually published their preclinical work. And these are just some of them shown here. MPA data is included in all of these publications, and as I mentioned, a lot of these have been successful and are currently in the clinic. So now let's get into what exactly the Membrane Proteome Array is. The MPA, for short, is a library of 6,000 human membrane proteins, all expressed in unfixed cells. In short, the proteins are arrayed into 384 well plates, one protein per well, in plasmid format. We then perform a transient transfection into HEC293 cells, such that all the cells in a well are overexpressing one particular membrane protein. We then take the customer's antibody and screen it across the library using flow cytometry methods to measure binding. So it is very sensitive, quantitative, and we can maintain the transfected cells in unfixed conditions. The result is a specificity profile of each antibody so that we can assess if there are any off-target interactions. Here is what you can expect from a standard MPA project. The customer simply needs to send us 500 micrograms of each antibody to be tested. All antibodies undergo these 3 steps in the workflow: assay setup, MPA screen, and hit validation. The assay setup is an optimization step where we test titrations of each antibody against appropriate controls in order to select optimal screening conditions. Once those have been selected, we will then screen the antibody against the 6,000 protein MPA and see what hits come up. All hits identified in the screen are then subjected to a validation experiment, wherein we will cherry pick out the hits, perform a new transfection, and then test titrations of the antibody against just those hits to confirm that they are real interactions. In order for a hit to be validated, it must exhibit signal above the background and also dose response to indicate that it is a specific interaction. The final deliverable for the customer is a detailed report including all the data for every antibody tested. All 3 steps of our workflow are performed by flow cytometry using unfixed cells. Timelines can be as little as 4 weeks, and we also offer some optional post-project benefits, such as clone transfer or enhanced validation studies. The MPA is the largest and most comprehensive collection of membrane proteins. The 6,000 proteins cover over 94% of the human membrane proteome and includes a diversity of proteins. Our library also includes over 200 heterocomplexes, such as alpha-beta integrins or TCR complexes. And we are also in the final stages of developing a secreted proteome array, and we do expect this to be available to our customers soon. Since the MPA is a flow cytometry-based platform, it is very compatible with many biotherapeutic modalities, including IgGs, SCFV, bispecifics, VHH, and even peptides. We have also worked with numerous CAR-T companies to screen their molecules. While there are different methods that can be used to screen CAR-T therapeutics, including cell binding and functional assays, we have found that the most efficient and highest quality data is obtained by screening the receptor portion of the CAR in an SCFV format. Our CAR-T customers have found that this method is ideal for successfully demonstrating specificity in their IND submissions, and many of them have been successful and are currently in the clinic with their CAR-T molecules. There are a number of benefits that are unique to the membrane proteome array, and I will talk about some of them in the next few slides here. One of the benefits is that we have validated the expression of the MPA library in 2 different cell lines. While hex cells are standard and are ideal for expressing human membrane proteins, there are cases where hex cells can result in high background levels. For example, screening an antibody against B7H3 results in high background levels because B7H3 is endogenously expressed on hex cells. In these situations, the hex cell data is unusable, but we can pivot over to a different cell line called QT6. QT6 are quail cells, and the benefit of using them is that they are evolutionarily divergent from mammalian cells to the point that we can greatly decrease background levels. And as you can see on the right-hand graph, now we can easily distinguish the B7H3 binding. The MPA is the only specificity testing platform to offer an alternative cell line for screening, and we have been able to rescue many projects that otherwise would not have been completed due to background issues in hex cells. Perhaps the greatest benefit of the MPA platform is that the entire process is performed using unfixed cells. While developing the MPA, it was very important for us to develop it in a method that would preserve native epitopes. Fixive reagents that are often used in tissue studies or other specificity testing methods can alter the cell and membrane structures in such a way that native epitopes are obstructed or unnaturally exposed. This can result in false negatives or false positives, neither of which are ideal because of the risk of a false negative or the additional work that would be required to track down a false positive. To demonstrate the differences between screening on fixed versus unfixed cells, we performed a head-to-head experiment using a CD19 antibody. On the left-hand side, we tested the antibody on the MPA using our standard methods of unfixed cells. You can see that an off-target interaction was identified. However, when we screened the same antibody, but this time fixing the cells first before applying the primary antibody stain, you can see now on the right-hand graph that the CD19 binding was unaffected, but this off-target that we had previously identified and validated was consistently lost in the noise. Most likely, this protein was more sensitive to fixation and its epitope was affected in some way. You can see how this fixed cell screen can be very misleading, causing you to believe that the CD19 antibody is specific to its target, when in fact we know that there is a concerning off-target. I would like to take a few minutes to discuss the use of MPA data in IND filing. Specificity testing is a requirement for IND filings, and the FDA's guidance from 1997 advises using human cells or tissue cross-reactivity studies to assess cross-reactivity. I would like to think that they recognize the fast-changing pace of drug development and the technologies being used, as they also advise that appropriate newer technologies should be employed as they become available and validated. We are now certainly seeing that shift as cell-based protein arrays like the MPA have become more popular. From surveying our customers, we are seeing the success of the MPA in a number of ways. Over half of our customers have used or are planning on using MPA data in their IND filing. Of the ones that have already submitted, we also know that a number of those submissions were successful with only MPA data and no tissue cross-reactivity data for both antibody and CAR-T modalities. We have a global customer base, so we have seen that MPA data has been used in successful submissions to multiple regulatory agencies, including the FDA, EMA, and NMPA. And most interestingly, 25% of surveyed customers reported to us that the FDA specifically requested them to test their molecules in a newer array-based technology like the NPA, and then resubmit their application. I think this really points to a shift in how the FDA is recognizing the importance of protein arrays for assessing safety and is really changing the landscape here. This helps lead me into my next slide to discuss a very exciting development for us. Late last summer, our MPA platform was the first technology to be accepted into the FDA's pilot ISTAND program. ISTAND is an acronym that stands for Innovative Science and Technology Approaches for New Drugs. What it means is that we are working closely with CDER and CBER to formally qualify the MPA as a new drug development tool, which would really set it as a new standard at the FDA for IND submission and assessing the specificity of a molecule. So as we are working with them, the FDA is providing input on the MPA, including our methods, reporting, and QC metrics. We are actively working on the qualification plan and hope to receive the formal drug development tool designation in the next year. So how can you work with us? We recognize that different companies would like to perform off-target specificity testing at different stages of their development process. We offer 2 service packages to meet those needs, whether you would like to test earlier in the process at the lead selection stage using our using our standard MPA service, or testing your final lead with us at the IND submission stage using our MPA plus IND service, which does come with some of its own additional perks and special benefits. To summarize today, there are a number of reasons that the Membrane Proteome Array is the leading specificity testing platform on the market. The comprehensive membrane protein library and cell line options are huge advantages, but what I hear repeatedly from customers is that the greatest benefit of the MPA is the use of unfixed cells in a flow cytometry detection method, and this is what truly sets the MPA apart from other platforms. Not to mention the quick timelines and flexibility of the MPA means that we have successfully screened over 1,000 molecules to date, with more coming in each day. Other than the specific benefits of the MPA, you also get to work with Integral Molecular. We have long been a leader in the antibody and membrane protein field, with over 20 years of experience in working on discovery and characterization of antibodies against the most complex proteins. We are trusted by over 500 companies and also have partnered projects with multiple companies such as AstraZeneca, Merck, Daiichi Sankyo, Ono, and Context Therapeutics. In addition to the membrane proteome array, we also offer products and services for antibody discovery using our MPS antibody discovery platform, antigen presentation using our lipoparticles, and conformational epitope mapping using our shock immunogenesis epitope mapping service. To find out more information about any of our other capabilities, please feel free to go to our website where you can learn more about each of our platforms. Thank you again for joining the webinar today. I would be happy to answer any questions during the Q&A portion of the webinar after all of our speakers have gone. But for now, I would like to pass it on to our next speaker.
Hello everyone, thank you for attending this webinar. I want to start by thanking Integral Molecular for hosting this, as well as inviting me to speak about the experiences that Cabaletta had using the MPA technology for de-risking their CAART product candidates for IND filings. Here are my disclosures. Let's start by understanding the technology that Cabaletta is pursuing in order to generate the CAA CAR. It's sort of a twist to the traditional CAR T product. On the left here, you're looking at a Kymriah CAR, which is clinically approved right now for treating patients with refractory and relapsed follicular lymphoma. So here, a patient's T cells are harvested and armed with a CAR molecule that contains an anti-CD19 SVFV as a targeting domain, along with 4-1BB and CD3 signaling domain. Once the T cell expresses this CAR, it in turn binds to all CD19 antigen expressing cells in the patient's body, eliminating any cancer cell that express CD19 along with healthy B cells which are known to express CD19, resulting in a patient that has no antibody mediated immunity left. However, Cabaletta has modified this technology by removing the autoantibody fragment as a targeting domain and replaced it with an autoantigen, resulting in their CAA CAR T cell. Due to the presence of autoantigen, the T cells specifically engage with pathogenic B cells which are expressing autoantibodies against this particular autoantigen. This approach allows us to conduct a selective and a specific elimination of pathogenic B cells in an autoimmune setting. So depending on the autoimmune indications, the targeting domains can be swapped with their respective antigens to counter the autoantibodies that are being produced within the patients. Currently, Cabaletta Bio initiated 2 clinical trials using this CAA CAR technology, which is a DSG3 CAR T therapy for treating patients with pemphigus vulgaris and MuSK CAR T therapy for treating patients with MuSK-dependent myasthenia gravis. In the respective indications, the autoantibody titers have against DSG3 and MuSK drop after rituximab treatment. However, it is sort of incomplete elimination of these pathogenic B cells, and these autoantibodies tend to persist during the relapse. So to counter this problem, Cabaletta has designed these 2 CAR T products to treat the patients with the particular PV and for myasthenia gravis. Here, what we have done is that we've utilized the first 4 domains of DSG3 as a targeting domain along with 4-1BB and CD3 signaling. EC5 was eliminated because it's been shown in the literature that EC5 antibodies tend to be non-pathogenic. However, for MuSK, we actually utilize the full ECD as a targeting domain. So both of these AA CARs can now specifically target autoantibodies against DSG3 and autoantibodies against MuSK. Before we could finalize our lead CAR candidate, we needed to conduct an off-target binding analysis. Since most of the CAR that are used in the clinic right now use an antibody fragment, so the intrinsic risk of off-target binding is low due to the specificity of scFvs. However, in our case, we were working with 2 distinct proteins, and at that point, they could have multiple binding partners, binding domains, and it would be important for us to see that if there is an off-target binding that's happening. So along with our thinking during our pre-discussion, pre-IND discussion with the FDA, they also advised us to conduct a similar screening, right? They wanted us to screen our CAR T for off-target binding using either tissue cross-reactivity screens or a plasma protein array, as whatever is feasible for us. The toss-up was between tissue cross-reactivity study and membrane proteome array. We decided to go with MPA as he had it had expanded breadth and known capabilities of accommodating screening of non-antibody proteins, which would be ideal for us. So multiple companies were shortlisted for this task. However, we ended up choosing Integral Molecular. I will definitely talk more about why we chose them if it's not clear in the next few slides. But let's just jump into one of the studies that we did with Integral Molecular right now. All right, so let's look into the case study that talks about understanding the off-target binding analysis using the MPA technology for our MuSK-CAAR CAR program. Here, in order to utilize the MPA, we had to first generate a FC-tagged MuSK protein. So in order to do that, what we did is we kept the extracellular domain of MuSK the same. So the targeting domain that we had put on the CAR is exactly the same in here. And instead of the signaling domain, we switched it and an FC tag region right here. So what that did is allowed for solubility of the protein and purification. So on the right here is the schematic of this protein array, where cells are expressing in certain combination about 6,000 native human membrane proteins. They are scanned against the MuSK-FC protein that we have using flow cytometry, and if there's any specific target or binding picked up, you're able to get the analysis at the end of the study. All right. Let's do a deep dive in understanding how the MPA technology helped us de-risk our off-target binding for the MuSK program. Initially, before conducting a pan screen, we need to establish and set up assay parameters. What I mean by the assay parameters is that we need to know exactly what concentration of FC protein we are going to utilize for the pan screening in order to find optimal off-target binding along with the lowest possible false positive rates. In order to test that, you can see that Integral Molecular first tested two different cell lines, 293s and QT6s. Since both the cell lines were overexpressing protein A, the FC protein that we provided should readily bind to protein A. And you can see in the line in blue here is 293s show a much higher MFI upon binding with protein A compared to QT6s. So we ended up using 293s for further testing. If you look at the table on the right, you can see that 5 microgram per mil of the MuSK-FC protein should be utilized for further testing because it also has very low level of false positive rates, about 0.5%. So we ended up using 5 microgram per mil of the MuSK-FC protein for screening. Right, so here is the data that we received from our initial screening. The graph on the left has the y-axis showing the normalized binding, and on the x-axis, each of the dots represents one of the 6,000 proteins tested. Anything below the dotted line is considered to be a non-binder for the MuSK-FC protein. Anything above the line is considered a potential binder. So as you can see, 4 out of the 6,000 proteins were shown to be potential binder for the MuSK-FC protein. NF-FFR2, Nectin-4, MPC1L, and MMP16. None of these proteins, which are considered for a potential binder in the MPA screen, were ever implicated in the literature as being a binding partner for MuSK. To confirm the initial screening, we did a comprehensive secondary validation screen where only MMP16 was identified as the sole binder of MuSK-FC protein. So in the following slide, I'll show you how the secondary validation screen is conducted by Integral Molecular. All right, so here is the validation assay. Let's look at each of these individual components very carefully. It is very important to employ the right controls for interpreting this data. So solid lines here depict the binding affinity to the MuSK protein, whereas the dashed lines show binding affinity to isotype control. In our case, it is PD1-FC protein. It will basically show any non-specific binding to just FC region of both of these proteins, if a particular protein binds with both of them. So both FCs bind really well to protein A, confirming that the assay is working well. So you can see in the blue line, both solid and dashed lines have concentration-dependent binding to protein A, which is good. To distinguish between the two chimeras, we actually utilized another protein called 4A3. So 4A3 was expressed in cells which is actually an SCVFP, an antibody fragment that is known to bind the musk protein. This will allow us to further distinguish between the two chimeras. So we want to know what is specifically binding to the musk region versus just PD-1. So you can see here in the solid green, you also see a concentration-dependent binding affinity to musk protein, just like you're seeing with protein A in blue. So this shows that the assay is working well. We have a protein that binds specifically to musk and not to PD-1. We also have a protein that binds to both FC region itself. And here you can see, if you actually look at this graph itself, you would be able to say that MMP16 is nowhere to be found. It might not be a positive hit. But if we zoom into the data itself, so we are zooming into this lower region right here, you're able to see that if you follow the orange line itself, both at 20 and 5 microgram per mil, you see about twofold increase in MFI values in MMP16, this orange line, between the musk FC versus the isotype control. So according to the IM, or intermolecular validation acceptance criteria, we accepted that as a positive control because it was twofold higher than its isotype control. But as you can see that the data itself, or those orange line overlaps the vector-only isotype control, suggesting that it's a very, very weak binding within the noise levels, or potentially, as I said, very extremely weak binding. So we decided to follow up on that. And the follow-up studies basically showed the same thing. So we did an initial primary cell screen where we found that MMP16 was not a validated hit, discerning all the safety concerns. But again, you can clearly see that MPA provides such high quality data in terms of the report and the dissolution of these targets as sort of off-target binding. On a side note, I would also like to mention that Cabaletta recently licensed a CD19 binder from a company called IASO. We were happy to see that when we were doing due diligence, they also had utilized the MPA technology to de-risk their off-target binding effects. So this just goes to show how widely accepted and popular the MPA technology has been in the field of CAR-T. And as I talk more about the MPA technology and our relationship with Integramolecular, we have been collaborating with Integramolecular since 2018. I personally joined the company in 2019 and have conducted multiple studies with them. Cabaletta utilized MPA to sort of de-risk several of their CAAR CAR programs, and then we have used those reports for our IND filings. From an IND standpoint, we observed no pushback from FDA. They have, in every instance, seemingly accepted the data without any issues. Integramolecular has made several advancements to the MPA technology over the years since I've worked with them. As a matter of fact, recently we had a problem generating an FC protein for one of the programs, and they sort of provided us with different options by allowing us to use different tags. This allowed us to purify the protein wall and complete the study. So as I said, like, along with the assay turnaround time to be being very quick, the biggest thing about Integramolecular is the fact that the data that we get is high quality data, and then the study reports can be completely taken and added as an attachment in our IND filings. So it's always been a dream working with them. It's a very innovative and collaborative team, and I'm happy to see how they have taken this MPA technology and now basically utilizing that as the standard way for all the FDA approvals going forward, or de-risking studies going forward. With that, I'm happy to take any questions. And again, thank you everyone for joining this webinar, and thank you for listening to me.
Thank you very much for inviting me to participate in this webinar. The story I am about to tell you is about a project to develop a chimeric antigen receptor T-cell therapy for a rare form of thyroid cancer known as medullary thyroid cancer, for which there exist no cures. In the course of developing this CAR-T therapy, a pair of antibodies were developed to target a novel cell surface receptor on the cancer tissue, and the use of Integramolecular's Membrane Proteome Array turned out to be a critical tool in helping us select which antibody to move forward with in our preclinical work, regulatory submission, and then eventually to use with human patients in a clinical trial which recently opened. Here at the University of Pennsylvania's Perelman School of Medicine, I've been involved in clinical work as well as basic and translational work for nearly 40 years. As a transfusion medicine pathologist, my clinical work has involved running the blood bank, the apheresis unit, and hematopoietic stem cell lab at the Hospital of the University of Pennsylvania, as well as directing the manufacture of CAR-T and other cell therapies for dozens of phase 1 first-in-human clinical trials within our Center for Advanced Cellular Therapies, where the first successful use of CAR-T cells for leukemia had been demonstrated in 2010. At the same time, over the years, I had been conducting basic and translational research on the university campus that developed antibody engineering methods known as phage display to study human auto and alloantibodies that arise in a number of disease settings. It was not until this medullary thyroid project came along that my clinical and research worlds came together. I originally was introduced to phage display in the early 1990s through a paper that described how to make libraries of antibodies expressed on the surface of filamentous phage particles through linkage of antibody fragments to the gene 3 coat protein of phage. The first author of this PNAS article, Carlos Barbas, was a postdoc at Scripps Research Institute at the time, and we quickly became friends and scientific colleagues in this new field of antibody engineering. Shortly thereafter, we started a course on phage display at Cold Spring Harbor Laboratories in New York, where we would hold a 2-week lab course teaching groups of 16 students per year how to make phage display libraries and isolate antibodies from them. Along with Greg Silverman, a colleague at UCSD and then NYU, and a number of guest lecturers, including George Smith, Christoph Rader, Chris Norin, Dev Sidhu, and Ian Wilson, we did this annually for over 2 decades. Then, unexpectedly and unfortunately, a few days before the course was to begin 1 year, we received an email from Carlos which informed us of the following. He wrote, "I won't be able to attend the course this year. I just got the news of the discovery of several tumors in my neck area. Still don't know type yet. I hope I can call on you both for advice." Carlos then underwent surgery, and the tumors in his neck turned out to be a rare form of thyroid cancer known as medullary thyroid cancer. Unlike common forms of thyroid cancer that are derived from the follicular cells of the thyroid gland, the cells that synthesize thyroid hormone, MTC originates from parafollicular calcitonin-producing cells, also known as C cells. MTC comprises about 3 to 5% of all thyroid cancers, and long-term survival varies from 75% if the tumor is localized to less than 40% if the tumor is locally advanced or metastatic. The prognosis is associated with post-operative calcitonin doubling times in the blood. Less than 25% of patients are alive in 5 years with calcitonin doubling times of less than 6 months. And unfortunately, Carlos's calcitonin doubling time was 2 weeks. MTC is usually caused by mutations in RET, and in Carlos's case, his disease was so aggressive because his RET mutation consisted of a deletion of 2 amino acids that caused covalent disulfide bonds to be formed between adjacent RET molecules, resulting in continuous expansion of the calcitonin-producing cells. Given the dire nature of Carlos's prognosis, a number of us here at the University of Pennsylvania and at Scripps wondered if we could come up with some therapy for Carlos. Given my background in the manufacture of CAR-T cells, I wondered whether we might quickly create a CAR-T cell therapy for medullary thyroid cancer. The idea would be to identify a target, make an antibody to the target, and then make CAR-T cells that use that antibody fragment. In terms of the target, it could be specific to Carlos's tumor or specific to a target on any patient's medullary thyroid cancer. But the target could simply be something that would not only be present on medullary thyroid cancer, but also on normal C-cell tissue, since all normal C-cell tissue would have been removed during a patient's initial thyroidectomy. The overall experimental approach is depicted in a cartoon here that was eventually published a couple of years ago. It starts off with the identification of a potential target using RNA-seq, making antibodies using phage display against the target, using those antibody fragments to make a CAR-T cell, testing the CAR-T cell in vitro, then in an animal model, and then hopefully in Carlos. Analyzing a portion of Carlos's tumor obtained following his surgery by RNA-seq revealed a large number of transcripts encoding proteins that were more highly expressed in Carlos's tumor than in other tissues of the body. At the top of the list was calcitonin A, which made sense, though not useful as a potential target, as calcitonin is not a cell surface protein. Next on the list was something called GFR alpha 4. But what was GFR alpha 4? In this cartoon, GFR alpha 4 is depicted in red as molecules that bring together red molecules, depicted in blue, when the ligand Persephone, in green, binds to GFR alpha 4. GFR alpha 4 is a member of a family of cell surface receptors known as glial cell line-derived neurotrophic factor receptors, along with other family members GFR alpha 1, alpha 2, and alpha 3. The question at this point was, how specific is GFR alpha 4 to C cells? From previously published studies conducted a decade earlier, endpoint PCR had demonstrated that GFR alpha 4 appeared to only be present in the human thyroid gland and in none of the other tissues shown here, including those of other endocrine organs, the central nervous system, gastrointestinal tissues, and others. We then performed staining of tissues or cells for GFR alpha 4 transcripts. Shown here at the top shows the presence of GFR alpha 4 mRNA in the parafollicular cells of normal thyroid, as well as in MTC case 1, which is Carlos's tumor, as well as in MTC case 2, another patient with MTC. TT cells represent a commercially available MTC cell line from which we made a pellet, fixed, embedded, sectioned, and stained for GFR alpha 4. TT cells also demonstrated the presence of GFR alpha 4 mRNA. We then performed a series of in situ hybridizations to look for the presence of GFR alpha 4 transcripts in non-thyroid tissue and could not find GFR alpha 4 anywhere other than in thyroid tissue. Having identified a potential target for MTC, the next step was to isolate antibodies to it. Using methods I had learned from Carlos years earlier, antibody phage display was used to make anti-GFR alpha 4 antibodies. By starting with a large naive rabbit phage display library contributed by Christoph Rader, a former postdoc of Carlos's and then professor at Scripps Research Institute, we panned the library against polystyrene microwells coated with recombinant GFR alpha 4. In order to avoid the isolation of anti-GFR alpha 4 antibodies that might cross-react with other GFR alpha family members, high concentrations of soluble GFR alpha 1, alpha 2, and alpha 3 were used to absorb out potentially cross-reactive phage. From the panning campaign, 2 unique antibodies named P46 and P410 were isolated. Shown here is an ELISA showing the binding of soluble P46 and P410 Fabs to both isoforms A and B of GFR alpha 4. The question now became, how specific to GFR alpha 4 are these 2 antibodies? This is where the integral molecular membrane proteome array became an essential tool. When each recombinant antibody was run on the membrane proteome array, we discovered that while antibody P410 appeared to be exquisitely specific for GFR alpha 4, antibody P46 appeared to cross-react with SYT2, known as synaptotagmin 2, a synaptic vesicle protein in the central nervous system, certainly not something we would want to target in the brain. Based on the integral molecular membrane proteome array, all of our subsequent preclinical work focused on GFR alpha 4 binder P410. We created a single-chain FV construct of the heavy and light chain variable regions of P410, which we then incorporated into our usual CAR-T vector that comprises a 4-1BB signaling domain and a CD3 zeta chain of the T-cell receptor complex. Lentivirus made from P410 vector was used to transduce human T cells, and expression of the P410 CAR was detected with a Dunkey anti-rabbit fluorescent antibody, as shown on the top right. To be used in our experiments as a negative control were human T cells expressing a CD19-directed CAR, whose expression was demonstrated with fluorescent goat anti-mouse, as shown to the left. To investigate the signaling ability of P410 CAR T cells to react to appropriate specific and nonspecific stimuli, Jurkat reporter cells were transduced with P410 lentivirus. Jurkat reporter cells express GFP when activated through the CAR. Shown here in the red box are the activation profiles of P410 CAR T cells when exposed to the stimuli shown in the green box. As can be seen, P410 CAR T cells are activated by the MTC cell line TT, another MTC cell line MZ-CRC1, and by immobilized recombinant GFR alpha 4 protein, but not by K562 cells expressing CD19 or by immobilized recombinant GFR alpha 1, alpha 2, or alpha 3 protein, as would be predicted from the integral molecular proteome array. Conversely, in the red box here, CD19 CAR T cells are only activated by K562 cells expressing CD19. OKT3 antibody serves as a positive control for both types of CAR T cells. Media alone does not activate either CAR T cell, and non-transduced cells designated NTD are only activated by OKT3. Our next step was to determine whether human T cells which express P410 CAR T are able to kill MTC cells in vitro. As a function of effector to target ratio, shown here is the lysis by P410 CAR T cells of T cells and K562 cells expressing GFR alpha 4 isoforms A or B, but not K562 cells expressing CD19. The panel on the right again shows killing by P410 CAR T cells of GFR alpha 4 expressing K562 cells, but no killing of K562 cells expressing GFR alpha proteins 1, 2, or 3. Our next question was whether these P410 CAR T cells might have activity against MTC in a mouse model of the disease. Tragically, at this point, Carlos unfortunately passed away. Though it only took a period of about 3 months to identify GFR alpha 4 as a target for MTC, create antibodies to it using phage display, and develop CAR T cells that kill MTC cells in a dish, Carlos's disease was too aggressive. We nevertheless continued the work with funding from Carlos's family and was able to show that the CAR T cells worked in vivo in a murine model. Mice were given subcutaneous injections of TT cells and intravenous infusions of 1 million P410 CAR T cells 9 days later. Shown here in the middle 2 panels for each mouse is the elimination of the TT cell tumors by the P410 CAR T cells as measured by bioluminescence and by tumor volume as shown on the right. Here, mice were infused with CD19 positive, GFR alpha 4 positive NALM6 cells, and it was shown that tumor cells would be eliminated with P410 CAR T cells just as well as with conventional CD19-directed CAR T cells. The data presented here, along with data from additional experiments, were submitted as an IND application to the FDA in April of 2021, and IND allowance was granted about a month later. Integral molecular membrane protein array data were essential components of the IND application. A clinical trial at UPenn for MTC was put together by UPenn oncologist Roger Cohn and approved by the UPenn IRB and opened in May of 2021, coincident with receipt of IND allowance. The trial is currently enrolling patients, and patients undergo apheresis at my apheresis center at the Hospital of the University of Pennsylvania, and the patient's CAR T cells are manufactured in the UPenn Clinical Cell and Vaccine Production Facility that I direct. For those who are interested in learning more about the trial, it is listed as NCT 048777613 on clinicaltrials.gov. I would like to acknowledge members of a number of UPenn and Scripps laboratories, as well as our IND group within the Center for Cellular Immunotherapies led by Carl June. Also clinical trial PI Roger Cohen, Integral Molecular, and our sources of funding. Thank you very much for your attention.
Thank you so much to Rachel Fong, Darshil Patel, and Donald Siegel for those wonderful presentations on Integral Molecular's Membrane Proteome Array platform. We are now transitioning into the Q&A section of the webinar, so please submit your questions into the chat box. All right. Hi, everyone, and welcome to the live Q&A portion of this webinar. Please make sure to submit any questions you may have for the panelists now in the chat box. My name is Jonathan Grinstein. I'm Senior Editor at Genetic Engineering and Biotechnology News. I wanted to thank Rachel Fong, Darshil Patel, and Donald Siegel for those wonderful presentations on Integral Molecular's Membrane Proteome Array platform. And with that, we'll get started with the Q&A. Here's a question I have for Rachel. How often is it that MPA data can replace TCR data completely?
That's a really good question. Something that people ask a lot. And, what I can speak to is certainly more anecdotally from what customers have come back to let us know. And maybe Dawn and Darshil could chime in too with their own experiences, since they have that a little bit more direct interactions with the FDA through their work. But from our experiences, we have heard from a number of customers that have worked with us and included the MPA data in their IND submissions. We do know that there are many examples of customers that have been able to submit with just MPA data and not necessarily tissue cross-reactivity studies. I am not here to say that everyone will be successful that way. I think at the moment, since this is sort of a slightly newer method compared to tissue studies, which have been a standard for a long time, that it can depend on different things such as your therapeutic, your modality, whether tissue cross-reactivity studies were able to be validated for your molecule. So the short answer is yes, we know that it is possible, though it may not be as universal yet. And I can maybe throw it over to Dawn and Darshil if they have any comments on their experiences there.
I could, can you hear me? Yeah, yes. So I could just say that in our case, things were a bit different because this was a target that virtually no one ever heard of before. So doing tissue arrays with the antibody wasn't so much to see if there was an off-target non-GFR alpha 4 hit, but whether GFR alpha 4 was actually expressed on tissues other than thyroid. Right. So it's a bit different than like a CD19 thing where, CD19 has been studied for quite a long time. People know that it's not, on liver cells. So we did do, even before even thinking we'd ever submit anything for an IND, we did do tissue staining just because we wanted to verify for ourselves that GFR alpha 4 was specific to thyroid.
Yeah, I can pitch in as well. Can you hear me as well in there? Yeah, so for our case, I think we didn't have any issues just to make the MPA screen. So we actually just ended up going with the MPA data first to the FDA and did follow-up studies right after that. So we didn't require tissues.
I guess just to kind of follow up there, Dawn, just be clear, did you try doing other TCR for your molecule, or was it just the MPA data and you went to the FDA with that?
No, we did tissue staining with the antibody.
Right.
And submitted that. Yeah.
Right. All right. So it looks like you may want to go with more than one. So who knows? It depends on your situation. Here's a question from the audience. Can you leverage the technology to test the specificity of proteases?
I would say that's something we have not done before, but I think it's definitely worth a conversation if the asker can get in touch with us. It is a very flexible platform. Like you heard, it's flow cytometry based. The question is, can we actually detect a protease in our system? So that's something that we can discuss sort of, is it just a straight up protease? Is this some kind of modification that you have? We mostly work with antibody-based therapeutics, but we have done other small molecules and peptides as well. We just have to kind of talk with you to get a little bit more information about your molecule and what are the possibilities of working with it to see if it's compatible with our system.
It's a pretty broad question, but at what stage of antibody discovery or development should you test for specificity?
I feel like I can start off again and then ask Dawn and Darshil to chime in. So, for us, we our goal is to help drug developers really de-risk as early as they can. Typically, that ends up being somewhere around the lead selection stage, where maybe you have half a dozen candidates to really start weeding down to your top leads to just further develop until you hit that final lead. That's where we see a large number of our customer base, and I think it obviously depends on sort of what your own resources and development process is. That being said, we also have a large sector of customers that come to us at the final pre-IND stages with their actual final lead candidate, and we just test that one candidate in our IND service. So I think it really depends on sort of what kind of risk you're comfortable with, what your development plan is, and where you want to make those certain decisions. But I would say that a lot of people do like to come a little bit earlier on and come with a panel of antibodies. That way they still have some backups, and you don't necessarily run the risk of going in with your final lead and then finding out something that you may not want to find out at that point. Don Darshil, any thoughts there?
Sure. Yeah, so I've used the integral proteome array twice now for 2 projects. I talked about in my talk the first project, and so in the case of the first project, the 2 antibodies were discovered, right off the bat. The proteome array didn't exist yet, so we made CAR T cells out of both, and we did lots of experiments with both of these 2 antibodies. And it wasn't until we heard about the proteome array that we had the work done and realized that one of the 2 antibodies was probably not something we would want to pursue because it had this off-target hit. And so at that point, we did all of our work with just the P410 antibody. I think we actually put both of them in mice. A lot of the experiments I showed here, I think we carried both antibodies for a while, but that's just because we didn't have the proteome array. In the more recent project, for targeting a different something different, I generated 30 different binders. We made 30 different CAR T cells, and we looked at which CAR T cells in a mouse work best. And 2 of them we picked as the best, what we thought were candidates. And at that point, we had the proteome array done to show that both of them actually were fine in terms of not having off-target hits. And then we chose one of the 2 to pursue for some other reason. So I think the idea is, depending on your project and how many potential leads you have. Obviously, if you have 50 leads, you're probably not going to do 50 proteome array things. You may want to do some other experiments first to kind of narrow down to some reasonable number of candidates. So that's been my experience.
Yeah, and I can also chime into that. So I think when we started doing Membrane Proteome Array, I think we had already picked a lead candidate. So we had an idea, this is our lead. And because we were going with a CAA CAR, we already had like a protein that was identified. And we had some background of understanding of what it binds to. So when we did that, we identified another protein that we thought was going to be potentially there. But in our MuSK program, which I just talked about, we didn't expect MMP16 to be there. And we realized that as we start doing more programs with intermolecular, we're starting to switch our views towards going more toward the lead selection, this time frame. So we started to work with intermoleculars, give them more proteins or more binders initially, rather than going with at the end for the lead select after the lead is finalized. So I think my views are the same as, again, Dawn and Rachel. I think it depends on your project and the scope itself. But I think during the lead selection time, it would be ideal because you don't want to go put in all the effort and do all the work and then realize there's some sort of off-target binding and then redo and optimize your lead. So
kind of staying on that topic there, Darshil, so and specific to your project and off-target binding, how did you perform primary cell screens to investigate the MMP16 off-target interaction?
Sure. So I think, yeah, as I mentioned in the talk as well, that MMP16 was sort of we thought it was going to be a very low binder, but also was a surprise the fact that it showed up in the screen. So in order to do that, first thing we did is we tried understanding what MMP16 protein is and where it's expressed. So I think we identified over 7 different organs that had high levels of MMP16 expression. Using that information, we isolated and procured cell lines for those primary organs. We were able, I think we did about 2 to 3 cell lines per organ that we were testing out. And once we had the cells growing well, I think what we did is a cytotox analysis. So what we did is we co-incubated those cells, primary cells, with our CAR. And the idea was that if our CAR is binding to MP16, we should see some toxicity. So at that point, when we did the screen, and all the cells that we tested out, we did not see any toxicity. So it sort of suggested that there might be some sort of off-target binding, but it was very, very low. And also, as I mentioned in the screen itself, it was a very low NMFI. So we expected that results to be there.
I have another technical question, so I want to move over to Don. How did you format the P54-6, P4-6 and P4-10 fragment antibodies for screening on the MPA? And does the FDA request that the compounds screened in vitro contain the same linker and hinge domain that will be present in the CAR used?
So the antibodies were sent over to Integral Molecular as recombinant FAB fragments. The antibodies were discovered from a rabbit naive phage display, rabbit Fab phage display library. So they were Fabs, and that's how we had discovered them, and that's how we characterized them. And so that's how they were examined by the proteome array. The way they are in the CAR T cell is different. It's isolated variable light and heavy link with a linker. But that's not what was tested on the proteome array. We didn't create a soluble single chain FE protein. And the FDA, at least at that time, didn't seem to mind, or couldn't tell the difference. I mean, I don't know that they really even knew, what it was a Fab or yeah. So I don't know if things are different now, but back when we did that, the Fabs were tested. Obviously, if it's a single chain V, you probably need to have some tag on it as well to detect, which for Fabs, it was just a lot easier for commercial antibodies against Fabs for Integral Molecular to use.
I wanted to shift over to some questions about using this with the FDA, and we have one here from the audience. Do you know the FDA's stance on screening soluble FC fusion proteins versus full CAR T cells? And did you need to do validations with cells? Rashil, do you think you can take that one?
Sure, I can take that. So we hadn't had any issues. So the idea there is that when you do a full CAR on a cell or an FC protein, we're still trying to make sure that the extracellular domain that's expressed on the cell is still intact. So we try to make sure that the full protein, along with its hinge or along with the linker, was co-expressed with NFC. So at that point, the binding that is happening, off-target binding that's happening to the region that's exposed is similar from an FC perspective or from a CAR on a cell perspective. I think it would be nice to have a validation done in the cells directly, because that way it sort of alleviates the burden of creating an FC protein. But from an FDA standpoint, we haven't had any issues when we did the screen with the FC soluble protein.
And I'll also just chime in, we have two great case studies here from Don and Darshil. We also have many other customers we've worked with who end at the CAR T modality. Similarly, they screen SCFV FC fusions with us, and we know that they have been successful getting that through the FDA as well.
Here's kind of a question that everyone may want to know, but how does the turnaround time for a project, using the MPA for an IND work? How does that compare to like your standard project? I don't know if Rachel, you have some kind of general information on that. And
yeah. I can absolutely talk about that. So, we do have two services, depending on sort of if you're using us for lead selection or for a pre-IND service, as well as sort of all different combinations. that you can select. But our timelines, so the fastest timeline we can do is a 4-week turnaround from when you send us all the paperwork and your test molecules to when we deliver you the final IND report. So it's very quick, it's very fast. And then of course, obviously we're available for answers, for questions afterwards. And if you need any kind of revisions to the report, some companies just have very specifics that they would like. For our standard projects where you're kind of more in the lead selection stage, those projects can be about 6 to 8 weeks. It's also a very quick process, and in that time frame, we can actually test up to 8 different molecules. They can be against different targets. They can be different modalities. Basically, if you want to, you can mix and match different programs and send them all to us as one project. And within those 6 to 8 weeks, we can get you data for 8 molecules. You can certainly send more than 8. It will just expand their timeline a slight bit. But yes, it's a very quick process, and we have a very good track record of sticking to those timelines.
Okay. It's quite a few specific questions here about, can the MPA isolate a specific antibody against a target of interest from a mix of antibodies, and whether, yeah.
I can comment on that briefly if you want.
Yeah,
I think what this person is asking about is not so much specificity testing, but maybe more target deconvolution. If you have interesting phenotypic antibodies, perhaps, or you have some kind of platform where you are identifying antibodies in a different method as opposed to a target-specific discovery. The answer is yes. So you can see with this kind of MPA platform, we have it at our disposal that we can screen antibodies against it, not for specificity, but just to see what comes up at all. So we have done those sorts of deconvolution projects. If you go to our website, there is a case study on work we did with Toshin, which is now under ABSCI, where we helped them deconvolute some of their antibody targets. So yes, that is something we can do as well. If you get in touch with us, we can talk about the specifics of those types of projects.
Wonderful. With that, we've come to the end of our webinar. I'd like to remind everyone that this webinar will be archived on the GEN website. That's at genengnews.com. It'll be there for up to a year. So if you missed any parts of it, you can watch it again, or feel free to forward it to any of your friends and colleagues who you think might be interested in this content or the webinar itself. Most importantly, I'd like to thank Rachel Fong, Tashil Patel, and Donald Siegel again for this incredibly informative presentation and Q&A session. I'd like to thank you, the audience, for your attention and some really great questions. Make sure to follow up if you've got any more. And a very special thanks goes out to Integral Molecular for sponsoring this webinar. Hopefully we'll see you again at another GEN webinar in the near future. Goodbye for now.